Well, hello everyone. Welcome here to our new series, or restarted series of the New Researchers webinar. And today we have the pleasure of having Saha with us. He comes from India, used to work or still has a position as research scientist at the National Defense Laboratory in India, in the Defense Metallurgical Research Laboratory, and he has taken a sabbatical leave to join us here at INL. So I will leave you here, Saha, to explain what he has been doing regarding the eyeing the materials, isn't it the title of his talk. Thank you, Saha. Please.
Yeah, thanks a lot, Mariam, and good morning to all the attendees here. So, in fact, as Mariam said, that I am coming from India, and so I'll just want to tell a little bit about where I come from. So, so uh, yeah, so I work in the Defense Metallurgical Research Laboratory in India, and I've been working there in the Electron Microscopy group. And so I've been kind of mainly involved with characterization of different materials, and I have been, in fact, eyeing the materials with different techniques and different uh, length scales to gather different information, which is essentially important to understand the material. So without taking any further time, let me just start directly move on to what I'm going to speak about. So first, as in, I'd like to kind of uh, uh, tell a little bit about the dual beam equipment, which is used in the material research, because as in, this is one equipment that I have been handling personally, and I'm the facility in charge, and we can do a lot of things in material research using the dual beam equipment. Then I'll come on to the uh, characterization by different techniques, and then correlative studies by different techniques to gather more information about the material. And finally, as in, you all can kind of, with all that, you can understand what my research experience and my interests are. So I'll move on with the very first well-known example. Why do we need to eye a material? Why do we want to see a material? So if I just take some example, okay, these are two different things that we are seeing. One is, on the left, is a diamond, and the other one is a graphite. Now both are different when we just directly look at it. Now, why is it different? If we try to answer that question, we have to look at it at keep on moving closer to the material and keep on looking at different length scales. So once we move on from this, uh, because both are made up of carbon atoms, and but the difference is the way they are bonded together. So if you look at it, the atoms in carbon in graphite are different, which is on the right, and on the left side the bonding is different. So it comes on to the crystallography of the material that governs the what the final outlook or the property of the material will be, because diamond, as we all know, it's the hardest material, whereas graphite is primarily used as a lubricant material or other high temperature applications. But so why is it different? Now, in terms of processing, what happens? Uh, they exist. This is the phase diagram, simple phase diagram of the carbon, which shows the different phases which exist at different temperature and different pressure. So what essentially we have is we can process, or rather tune, our experimental conditions so as to derive one particular crystal structure that will give us some desired property. So essentially, these three things are interlinked. The processing, the property is processing to property is interlinked, because whatever processing conditions we'll get the final property of the material, and that in turn is linked to the structure of the material or the sample that we'll be studying. And all this is actually linked with the characterization. We need to characterize to understand the process. We need to characterize, or rather eye the material that I say, and study for the property of the material. and link it to the structure. So why do we need to look at it? So in general, I'll just start off with one simple example of a simple heterostructure material or a thin film as it is. As we can see that these thin films are, as in all the functional activities or functional device structures are generally in the form of multi-layered films. And each individual layer thickness, their composition, the defects, etc., actually govern the final property of the device structure that has been grown. So from the previous slide that I had shown, so here the growth to the final property is linked, and that has to be studied or rather looked at in detail in the cross-section mode because they are in the form of stacks of different layers together. So now coming to the first part, as in using the dual beam, how this can be done. So using the dual beam, we can very easily it, uh, see the cross section of a or a heterostructure. What we can simply do is, because of the typical geometry of the dual beam, the this is from the one that is there in my earlier lab, and here at INL also we have a high-end Helios system that is there. Using that, what we can do is essentially we can mill the mill or rather dig into the surface or and then image the surface, so we can directly image the cross section and its features. This is just one example highlighting that we can actually observe the different multi layers together. This is another example where we are trying to look at a cross section of a solar cell. So we can actually see all the different layers, and in combination with the EDS or the energy dispersive spectroscopy technique within the machine, we can actually identify the different layers together which are present. So whenever as in so this is a very simple tool to actually just identify the different layers with it is within the kind of typical resolution that we are looking for. If the resolution or the requirement is beyond that, then we have to prepare cross-sectional TEM samples. This is like another example of a very small gate structure. If you see here, that's the actual device of a HBT, and this is the small gates here. And the gate structure, so using the dual beam, we can actually prepare very site-specific cross-sectional samples, and we can essentially see the different multi-layers, how they're stacked together. And we can study about the interfacial roughness or dislocations. So those are the things we can do. This is another example of a high temperature coating characterization. So why essentially I resorted to the dual beam here was because the region of interest, the interdiffusion zone here is uh, lying about 200 to 300 micrometers beneath the surface. So prepared a sample exclusively from that site specific region and studied it. And we identified the different phases. So you in the TEM using the diffraction technique. So coming on to another example where the dual beam can be used is when we are trying to look at some very brittle samples. This is one example of a very brittle sample, and where I was trying to look at the high temperature superconductor. And as we can see here, it just simply cracks even while we want to prepare a lamella or a thin lamella. And then we studied that for the different subgrain structures and analyzed it. So this is another example where the dual beam proves to be quite useful because it can we can use it for preparing samples for studying in the TEM in a very nice manner. Then coming on to another example of what other things we can do. Like as I showed earlier that we can actually image a cross section. Now let's suppose we take a stack of cross sections together, we can actually see the entire sample in a three-dimensional manner. Like we can see here, we as we keep moving along the other direction, we keep seeing how the phases evolve in the third dimension. So, and this can this is just a video or animation to show that, but we can actually derive quantitative information by actually stacking these images together by using softwares like Amira and others. So you can actually find the grain size of different phases and the porosity existing in the material. So this is one example. Then the other example here is showing, like, we wanted to study the silicon sample exactly under a nano indent. So we can see here that there is a subsurface crack that has been generated and how that has propagated. So if we keep taking stacks of cross-section images, we can build a 3D microstructure or 3D tomography kind of a study we can do for different samples. Then the other thing that we can use it for is sample preparation for other techniques. This is one very high-end technique known as the atom probe field ion microscope, which takes in only needle-shaped samples. And those needle-shaped samples can also be prepared using this dual beam. So dual beam, using that, we can actually uh, pick up a small kind of a cylinder from the region of interest or the sample, and then we can weld it to the surface or a dummy needle and then thin it. And then in the final study, we can actually study for the using the atom probe, where we will be using a high voltage, uh, high voltages to field emit the uh, field emit from the tip of the needle the different ions and identify them and build the entire thing, entire structure in three dimension. And this technique actually has a single atom depth resolution, so you can actually know which atom is sitting where. So this technique is used for studying uh, nano clusters or uh, features which are like not even in a grain kind of it's within the subgrains or at the boundary, some nano clustering at the boundaries. So this is used there. So this is just showing how it is, as in it's we can kind of, after making the cylinder, we keep thinning it using the ion beam in a cylindrical manner to derive to a very pointed tip. The other thing that we can use the dual beam equipment is for preparing other samples for in situ testing. So here, this is one example showing that where I actually picked up a carbon nanocoil and fixed it uh, onto two pillars like this, and from the top the indenter is to come, and we derive, or rather we analyze for the strength of the carbon nanocoil. So this was done for the in situ nanomechanical testing. The other things that I've been working on are like nano patterning of device structures or prototypes. So this is one magnetic sensor that we did at DMRL, and the this prototype thing was fabricated using the ion beam. This is one simple capacitor structure that was fabricated. So in all, this is one application, prototyping. The other is for which I mentioned, critical samples, TEM studies for which are very site specific. Then for brittle samples, this is again another example showing brittle samples and a very site specific. This was a real study that we were doing on an aero engine fuel manifold, and uh, I was trying to look at why it has actually cracked. So as we can see, it the region of crack within this within here, and uh, if I look carefully, the region of interest here, there is some phases which have come in, and that is actually responsible for cracking of the material, or rather the component. This is at the component level when I'm talking about. So when I look at it, I looked at it and we found that it is actually some phase that has formed with the chromium getting segregated in the superalloy. This is from the superalloy, and so uh, chromium has got segregated, and that resulted in the cracking, or rather failing, of the fuel manifold of an aero engine. So all this that I was talking about, so in many of the cases we had to look at the cross sections, and we had to prepare lamella. So some uh, we prepare by the dual beam when it is like uh, very site specific requirement. Now if I don't have that kind of a site specific requirement, then we can prepare lamellae, or rather TEM samples, using the standard technique, which is the standard as in sandwich method, as it is called. So this is simply a schematic showing how we prepare cross-sectional samples, because uh, our requirement for analyzing the TEM is that the lamella should be electron transparent, or in other words, we want the sample to be less than about 150 to 100 nanometer in thickness. So how do we get that in a cross section mode? So as I was talking earlier, that the samples, or rather the real structure or a device structure that one will get, will be in the form of multi-layered. So what essentially we do is we first take the cut two pieces of that, and then sandwich them together with an adhesive, or and that adhesive needs to be electron conducting. It needs to be a conductive glue. Then after we do that, after making that assembly, we push that assembly inside a slotted rod, inside a slit, which has to be in the form of a circular rod that has a slit, and we push that assembly inside that, and that rod is then, to give it enough mechanical stability or strength, we push that inside a hollow rod. Now all this, as in at each step we have to put in the glue and leave it for curing at each step. Then once this final assembly is we get, we slice those. Now essentially, now if you look at this one single slice of it, what we see, we have the entire cross section features that we are interested in, but this, at the moment, the thickness is about 400 to 500 micron, but that is not what a TEM can take. So we need to prepare it further. So we mechanically thin it via different means and reduce it to about 100 microns or lesser, and then finally use ion beam polishing and reduce the sample thickness to less than 50 nanometers, or even zero, where a hole will be formed. And then we put it in the TEM and study for the features that we're interested in. Like we want to study individual layer thicknesses. So these are some examples. So this one is the solar cell that I had shown earlier, that was uh, in the ion beam image. Now this is in the TEM. So a cross section that has been prepared, and we observe all the different layers, and we know about the interfacial roughness, about their thickness, and the different contrast mechanisms that exist within the TEM shows us this kind of a contrast that we are seeing due to the even the composition. Then the second example is a distributed Bragg reflector for a vertical cavity surface emitting laser structure that we were studying. This is a gallium arsenide, aluminum arsenide, multi-layered heterostructure, and we're interested in studying the interfacial roughness. So this is just highlighting that the features of interest and how we are looking at those things. Now coming on to electron microscopy techniques that uh, I have primarily been using for all these different purposes. This is the first one that I would like to highlight is a bright field and a dark field microscopy. So with the bright field and dark field microscopy, what we can see are, this is an example showing a gallium nitride film being grown on sapphire, and it's the study in the cross section mode. What we see are these dark contrasts in the bright field image, or micrograph rather, are dislocations. And the dislocations we see, they are like starting from the interface and moving up as the growth would have as in continued. So, and these are showing two different modes of imaging. One is the bright field and one is the weak beam. We in the weak beam, so basically what is happened is, or rather in the imaging technique wise, in a bright field we are using basically the transmitted beam for imaging, and in the weak beam we are using the diffracted. So whenever the beam transmits through the TEM sample, we have one transmitted beam and the other ones are the diffracted beams. Now this, under the weak beam technique, not going into the details, but we are able to image the defects, or rather dislocations, in with a much higher resolution. That's essentially the, so now the second one that I'm showing here is the analysis of the dislocations. I can actually, we would have heard about the Burgess vectors of different types of dislocations. So under different imaging conditions, we are able to see different types of dislocations, and then we can find out about the dislocation density that is existing in the material. This is so since I was studying this for gallium nitride, I'm showing this is uh, the G0002, or rather the imaging vector is the growth direction here, and 112 bar 0 is perpendicular to the growth direction. So basically what I'm trying to see is the sample being imaged from different directions, and we are seeing different types of dislocations. Then the third is about the diffraction, the selected area diffractions. So if once we keep tilting the crystals, we can get the different, the sample would keep getting oriented differently with reference to the electron beam in the TEM, and we will have the different diffraction patterns. Now we can use that to map the entire reciprocal space, and we can derive at the crystal structure of uh, the particular sample that we're investigating. This is one example where we were unaware of what we were looking at. And this, we actually identified it to be an orthorhombic phase, which was in a high temperature coating material, and we identified that phase using the selected area diffraction and tilting by, or rather systematic tilting, that was carried out. Then coming on to the other technique is the convergent beam electron diffraction. This is again another technique which we can use to identify the polarity of different uh, polar material. This is showing an example of a gallium nitride uh, sample, and what we see here, this is the experimentally observed convergent beam electron diffraction pattern. And if we see the bottom one, we see just that uh, the intensities have flipped when we have moved from, uh, as in if we look at the reflections, the intensities have got interchanged, highlighting that they are actually uh, uh, they are in the reverse polarity. And this we can observe, this cannot be detected by other techniques or in the imaging mode. So this is one very specific technique and very sophisticated, and we can actually use this technique even for finding out the thickness of different lamellae at particular location using this convergent beam technique. So essentially, we've used for defect studies, for phase identification, and for polarity determination for polar materials. Polar materials are those materials which, in crystallographic terms, uh, will have different feature along a particular direction. Like here, this is gallium nitride that I have shown. Along one particular direction, it is slightly extended. So that is why the bond direction becomes critical. The gallium to nitrogen bond direction, that becomes critical, and that defines the polarity in these materials. This can be further extended to even other polar materials like silicon carbide and others. This is showing another processing property correlation, which uh, I'll be discussing. So this is in short the schematic of a gallium nitride growth process, and how and what is critical. So basically processing, showing the process parameters at different steps, and then finally if the device structure has got these kind of defects, it was detrimental for the device. So it is essential to understand where those defects are coming from. So in order to do that, we need to study. So this was one of my studies and was part of my doctoral research work, that I studied the gallium nitride growth on sapphire and studied for different nitridation temperatures. And we essentially identified a nitrided layer, and uh, and that nitrided layer itself is about 1 nanometer thick. So understanding the crystallography, chemical nature, all at just about 1 nanometer thick is quite challenging. And using microscopy uh, techniques, we did that. So this is showing the selected area diffraction pattern, the combined diffraction pattern coming from the gallium nitride, sapphire, the nitrided layer, and uh, whatever is not matching, we attributed it to the nitrided layer. So this is in one particular orientation. So this is like 112 bar 0 orientation in a hexagonal uh, crystal system. We are looking at it, and this is a fractured pattern. So now if we kind of tilt the sample by 30 degrees and acquire the diffraction pattern, what we observe is there are the spots, and we can uniquely identify the spots which came actually from just the nitrided layer. And then we studied it in greater detail using high resolution microscopy, using XPS to identify the chemistry. Then uh, we found that it is actually a cubic nitrided layer phase, and that too, it is a rock salt aluminum nitride which is cubic. And this was published in the Journal of Crystal Growth and Design. This was part of my research work. Then coming on to some other works where we actually need to look at the material with different kinds of techniques to correlate the uh, final microstructure with the properties. This is, as we see, that this is showing one coating cross section. And this is again another complicated system that I was studying because of the mere large number of phases present in that. And this is the coating cross section, and it has got multiple layers and multiple phases. And I actually prepared site-specific lamellae from different regions to actually study. And here, this shows the coating cross-section at a particular uh, uh, this this region close to the interdiffusion zone. Then using the techniques of energy dispersive spectroscopy coupled with EBSD technique, we identified the various phases, their orientation relationships, and uh, defined as to what the coating cross section is and why that is giving us the high temperature oxidative reduction, oxidative protection to the niobium based alloy for which this was actually made. Then this is another example where I was looking at micro cracking in gallium nitride films grown on silicon carbide. This is showing uh, the Nomarsky optical microscope image of a micro crack that is observed on a gallium nitride film grown on. So I wanted to study why that is, why these cracks are developing in certain cases and why it is not developing in certain other cases. So we look at it in at different length scales. The first one, that same region, then I look at it in the AFM, where I get the very surface sensitive data from the this surface, and it shows clear hillock kind of structures the and the region of micro cracks that have generated along certain crystal planes. What we observe here, the red arrow shows the micro cracks and blue are showing certain other certain other type of features that are seen on the surface and which could have been formed by incomplete coalescence of the islands, or rather it seems like this gallium nitride was flowing as islands one over the other and due to incomplete coalescence. This is another technique that we to uh, use to study that same region in a different. This is using the electron channeling contrast microscopy, where we tilted the sample at very high angles and used the channeling contrast. We could even study, or rather see the dislocations on the surface, or rather dislocations being imaged which were emerging out of the surface and coming from beneath. So we could even study the dislocation density and count the number of dislocations on the surface. This is showing at the higher magnification where we could even see the growth spiral. So we were able to study how the growth had happened on the surface of gallium, or rather how the gallium nitride had grown on the silicon carbide. Now we wanted to see, we have already observed that there are cracks that have formed. Now, why those cracks have come about? So this was something which was quite interesting. We did a very high resolution Raman spectroscopy study on the regions the where it was showing cracking. So this is an optical image that is showing the micro cracks on the surface, and then this other Raman spectra map of the entire region. What we observe here are there are shifts. So now the shifts are coming because of the strain that is present in the gallium nitride film. And if we kind of give specific colors to the different ranges of these shifts in Raman spectroscopy, we get a map like this. So this clearly shows that at the region of the crack, the strain has been relaxed. Now, when so correlating that with the cross-section studies in the TEM, what we see here is there's basically the gallium nitride with the aluminum nitride interlayer and the silicon carbide as the substrate and showing the different dislocations. Then furthermore, what we can do is uh, we we study we studied the different types of dislocation using the weak beam dark field imaging, which I had mentioned earlier. We count the number of dislocations, we estimated the dislocation density from this technique, then from electron channeling contrast, even from AFM studies, and all of them we correlate to find that they are of the similar orders and they match. And even correlate it with the XRD studies from that gallium nitride film to estimate the strain in the gallium nitride film and also the dislocation densities, and they all come in uh, close orders. So this was one of the work that was recently published in the microscopy and microanalysis, where we correlated the strain measurements using Raman spectroscopy and correlated it with the XRD and the TEM. Then this is one of the most recent works that I was doing, and this is the work carried out at INL. So this we wanted to study the evolution of magnetism in molybdenum disulfide. This was one of the samples of, or rather, this is molybdenum disulfide, and we observe that it is a ferromagnet at room temperature. This is one of the desired properties, and I was trying to work for getting a room temperature ferromagnet, and this is one such sample. And this is showing the MH curve, or the magnetic hysteresis loop, that we say for the sample at different temperatures. And we clearly see that it is having ferromagnetic properties even at room temperature. And this work was done in collaboration with Manolo in the nanoscience nanomedicine group. Then uh, I went on to probe further as to why that is happening. So studied it with high resolution XPS, and we find that there are signals of iron in the material, which we were trying to dope. In fact, we were trying to dope uh, molybdenum disulfide, or make doped molybdenum disulfide to get magnetism in molybdenum disulfide. This is showing the XPS spectra of the iron 2p, and we find that there is the evidence of iron that has gone in, that is present within the lattice. This is the uh, in the high resolution TEM stem mode, and the EDS spectra. This is a colored map showing the presence of different elements, and we identify that okay, iron is present along with molybdenum and sulfur. This is just the high resolution stem Hadouf map, not a map, but a stem Hadouf, yeah, stem Hadouf map, and identifying that it is the molybdenum disulfide uh, sample that we are interested in. It uh, matches with the structure of the known molybdenum disulfide with the, and we can actually see the position of each atom along with its crystallography. This, when we blow it up and see at higher magnifications, we are even able to see certain regions where the intensity is lower than regular, and those we attribute to the presence of other dopant atoms. And uh, and if we do the electron energy loss spectroscopy in the TEM from this sample, we find the signals of iron. So we confirm that there is presence of iron in that, and that and other dopants, and that we have linked to the presence of magnetism. And this was actually presented at a recent conference in February. And this work is slightly ongoing work, and so I'd like to summarize by all my experience and expertise. So I mainly do TEM. I've been working with the dual beam, which we have used for many other sample preparation, then Raman spectroscopy, then the different classes of materials, gallium nitride, silicon carbide, all electronic materials, then other coatings, then oxides, other different thin films and functional materials. So I've worked on a range of materials, and my research interests are in, I look at application oriented materials. So starting from photonic materials to electronics and device structures. And I'm kind of the facility in charge for the two facilities at DMRL, the dual beam and the integrated Raman AFM system. So that's that's in short about me. And anyways, no work is complete without acknowledging the other people who have contributed in to all this work. So I'd like to thank my group head at DMRL and other sister laboratories for their collaboration. Some of the results that I've shown here. Then I'd like to thank our director DMRL and Government of India for granting me permission to come here and carry out research. Then I'd like to thank the FCT UT Austin project, under which I have worked on this 2D magnetic semiconductors. I'd like to thank uh, Leonard for uh, all his constant support and encouragement. And finally, I'd like to thank the others whom I have not been able to name them separately, but who have contributed in some form or the other to the result that I have shown here. So thank you all for uh, listening.
Well, thank you
Yeah,
very much, Saha. Thank you taking the time to prepare this nice presentation for us. We ran out of time, but maybe we can take one question if there's some pending question from the participants. They have a Q&A button here.
okay. Oh, there's no
Any
questions.
question?
Oh, yeah. Uh, how do you see the use of dual beam to study soft materials such as, okay, uh,
So the question is, how do you see the use of dual beam to study soft materials such as proteins or carbohydrate-based structures? From Miguel Cerqueira.
yeah,
Thank you.
yeah. Thanks, Miguel, for your question. In fact, the dual beam, because I'd like to say that the dual beam is meant for certain purpose, like as in if we want to look at some of the other different materials which are relatively hard, but soft materials I have looked at, like dental implants and other things, but I really doubt about the soft materials such as proteins and these, because even though the microscope, the newer microscopes allow you to uh, image them under an environmental condition uh, where you will be having environmental conditions such which will be under relatively lower vacuum levels, because all your materials such as proteins and those structures will disintegrate if you actually put it in a high vacuum mode. So uh, I'm a little doubtful as to if we are able to kind of uh, cure the material, or rather I'd say freeze the protein structure, then probably we can do something with that. We can freeze it using some epoxies or other things and then cure it. And so the position of the atoms or rather molecules remain there, then probably we may be able to look at, but directly the dual beam cannot be used.
We have a comment from Oliver, part of the IME's facility. One
Okay.
can embed soft materials like cells into a resin and stain them to provide enough contrast. There's a FibSem slice and view of biological materials.
Hmm.
Okay, thank you very much, Oliver. Thank you,
Yeah.
Saha. Thank you, Miguel, for the
Thanks,
question.
Oliver. Yeah.
And yeah, any further comments regarding what you've seen today, please contact directly Saha. As he mentioned, he's working with Leonard Francis. And in regards to the series itself, which will be happening every month, if you have any feedback or anything specific that you would like to comment about, feel free to send me an email or to the research office. So with that said, thank you once again, Saha. And to all in
Thanks.
Portugal, have a great 2 days off that will come up. Bye bye. Till next time.
Yeah.
Mm.